Hello and welcome back to a series of lectures related to botany, where today we'll be discussing the complete life cycle of an important freshwater green algae, that is Volvox. The Volvox is rated as the best known chlorophytian member, where its life as well as its dependence is controlled by freshwater, and within this freshwater it has a preference for those areas where the nutrient content of the habitat or the water is better, and where it has not to face competition in order to absorb water as well as the nutrients. These Volvox are usually seen in the form of a complex colony referred to as cenobium, and in the life cycle as a whole, every cell of a cenobium plays a collaborative activity to facilitate completion of life cycle as well as absorbing, using and excreting whatever it is producing as a waste or a byproduct. This Volvox is classified as a member of plant kingdom, that is Plantae, phylum Chlorophyta, class Chlorophyceae, order Volvocales, and family Volvocaceae. This plant or this algae has been seen to prefer those habitats where during the summer months the temperature is warmer and water remains consistently fresh. This in turn affects not only its survival pattern but also facilitates its improvement in density and diversity of the populations as well as of the community in which it is constructing or creating a part. Generally, it has been seen that wherever Volvox is seen growing as an independent population, its colors as well as its density and diversity induces modifications in the water color or water transparency, which is sometimes preferred in improving the ornamental value of the water bodies created within gardens and parks. It has been seen that if the habitat becomes contaminated, the density and diversity of the Volvox is getting tampered with, where their number, their structural characters as well as their morphological features are getting altered. Now, under morphological features, Volvox is usually classified as a colony. Now, colony refers to a group of cells which are adhered together either due to the presence of certain media or where they cooperate and collaborate with each other to facilitate the assemblage of large number of cells together to create one colony. Now, in case of the Volvox, the average number of individuals or Volvox individuals in that colony can vary from minimum 500 to maximum about 50,000. That means if the number is so variable, automatically there will be tremendous size variability of an individual colony of different species of Volvox. It is very rare when a single cell of Volvox is seen as an individual plant or individual alga. This is primarily because the interdependence of a group of cells in one particular colony facilitates not only survival but also helps it to synthesize what it requires for development, multiplication, reproduction, as well as sustenance of varied habitat conditions. Whatever the number of these different members in a particular colony are, it usually attains one uniform shape, that is rounded or circular. And in this circular colony, we have seen that usually every cell is having an ovoid shape, and at the peripheral portions, that is the apical portion, it is provided or it is facilitated by the presence of two flagella. We normally refer to this as a biflagellate algal cell. These two flagella facilitate the movement of individual cells as well as the movement of a colony of cells if they are adhered together or bound together. In each cell, we usually encounter that the peripheral surface is formed of the plasma membrane, and usually this plasma membrane is also on the peripheral outer surface protected by the algal cell wall. In some cases, we may have a cell wall developing around a huge colony, or in certain cases we may have individual Volvox cells having individual walls. In a colony, we have seen that once these cells adhere together, there is usually an intercellular connection. That is, one Volvox is connected to the next Volvox. Within one colony of 500 or 50,000 cell colony, we find that every cell is one way or the other adhered to each other, creating a complex structure. Further, once we look at these biflagellate cells' movements, that movement is also a collaborative movement. Collaborative refers to whatever direction a cell wants to move. Usually, each cell has a position to actually facilitate movement of each other towards that direction. The movement in a particular direction is actually dependent upon the movement by these flagella. That in turn reflects that be it colonial, be it single-celled Volvox, will find that these flagella actually facilitate its movement from one direction to another in the form of the flagellar movements. We find that within individual Volvox cells, there is a chloroplast, and that chloroplast usually shares two different shapes. One is referred to as a laminate shape, or sometimes it is also seen in a cup-shaped form. But irrespective of its shape, its role is identical. That is, it has to facilitate the synthesis of carbohydrates as and when this Volvox cell is getting exposed to sunlight. And in addition to this chloroplast, within each cell of this Volvox colony, we find that there is a single nucleus located towards one particular portion, preferring the basal part of the flagella. That means the flagella actually facilitate the normal maintenance of the nucleus, wherein whatever direction the nucleus directs the cell to move in, or whatever direction the nucleus facilitates the movement of the cell, the flagella will get or derive its direction from the nucleus itself. That is, nuclear manipulation is facilitating the movement of not only cell, but sometimes of the whole colony as desired or as directed by the nucleus itself. And in every Volvox cell, we also find that there is an eye spot, that is a directional spot located towards the filamental portion. That is, wherever we find these filaments, at that end we will also find an eye spot located in each individual Volvox colony, depicting thereby that these flagella and the nucleus is actually facilitating the cooperative activity within the cell of the Volvox and ultimately within the colony of the Volvox of a particular species, thereby facilitating not only the desired movement, but also shifting from the reproductive cycle sometimes towards the vegetative developmental cycle also. And once we compare the vegetative features of a cell which is active with the cell or with a particular Volvox with those which are inactive, that is where no metabolic activity is taking place, we see that in such cells there is minor morphological variation where not only the size decreases, that is, metabolically active cells are larger in size and inactive cells are smaller in size, but also we find that sometimes the flagella are getting retarded, that in turn affect the movement of these cells from one direction to another because of the absence of flagella. That obviously means that morphologically, whatever activities or whatever organelle are getting formed, those formations are under the control of the nucleus, which in turn governs not only developmental features but also governs the reproductive features of individual Volvox cells as well as the Volvox colonies. Then we come to the fourth important feature, that is the reproduction, which can be subdivided into two different parameters: asexual reproduction and second, sexual reproduction. Now, as the term indicates, in asexual reproduction, the multiplication of Volvox is not dependent upon the gamete formation, fusion of gametes, or the division of that zygote. Whereas in case of sexual reproduction, the developmental pattern of the gametes as well as their fertilization and subdivision is actually controlled by second phenomenon, that is where the control of genetic setup of the Volvox is facilitating the sexual reproduction also. Now, when we look at the asexual means of reproduction, that obviously means we have no gamete fusion, no division, or no formation of the zygote. But here, technically speaking, the vegetative means of multiplications are operative to divide one individual Volvox cell into many Volvox cells, which will ultimately result in the formation of a Volvox colony. Now, for this, obviously, one has to monitor the cell which gets transformed from active cell into a dividing cell, and during this morphological variations are also seen. Such metabolically active cells are showing first signs of their division by the gradual retardation of the flagella. That is, in every vegetatively propagating cell, we will first see that the flagella are getting retarded, and as soon as flagella get retarded, each of these cells, individual cells of the Volvox colonies, started to slightly enlarge in size and show swelling. That in turn indicates that within a colony, wherever we find such a cell which is slightly enlarged and has no flagella on it, depicts that it is now converting itself from the active cell into a reproductive cell. That is where it has to multiply to give rise to a progenial colony. In every such cell that gets actually identified as a means of asexual propagation, the enlargement of cell and dilution of the cell wall, that is the individual plasma membrane as well as the cell wall, is in turn facilitating the metabolic activity where the division, which is mitotic division, is taking place. Now, mitotic means where the parental cell generation will be comparable in terms of its morphological features as well as genetic features with what the progeny is, or ancestor and progeny will be identical. It has been seen that within one Volvox, sometimes more than 7 or sometimes even 15 cells can start entering the metabolic activity of division. That means we will have parental colony of say about 500 to 50,000 cells, and within this we may have about 12, sometimes even 15 cells now entering the activity of mitotic division, that is to create progenial colonies within the parental colony. Now, it obviously means that with this division, the parental colony will increase in size, and within this parental colony, we will have a large number of progenial colonies developing. And this division has been seen to usually complete in a short span of time, but is dependent upon two important features. One is where the substratum is of fresh water and is rich in inorganic as well as organic nutrients. That obviously means that the division is facilitated by fresh water and presence of nutrients within that water. And in addition to these, one more important feature is that the temperature at the time of division should be around 21 to about 24 degrees Celsius. That means the temperature is also facilitating the proper division of the parental cells into progenial colonies, and this in turn will facilitate replication of parental cells into progenial colonies, and from one parental colony we may sometimes have as high a number as 15 progenial colonies developing. Now, as soon as the desired number of cells are getting formed, that is where desired number is 500 or where desired number is 50,000, as soon as such number is getting formed, these cells in turn then try to protrude, that is move out of the parental Volvox generation, that is Volvox colony. And at this time, the parental wall of the Volvox colony ruptures, and these progenial colonies ooze out into fresh water, and at that time these have to actually show change in their curvature, because usually at the time of division, the flagella are found to develop towards the interior of this progenial colony, and for facilitating of that flagella to move out, this actually moves out, that is inverges from the interior to exterior, and that movement is converting now a dormant colony into an active colony, because flagella formation and flagella in turn facilitate the movement of these progenial colonies in the water from the parental colonies towards whatever habitat they prefer, or whatever habitat is suitable for their growth, for their development, as well as for the regeneration of the third progenial generation. Now, in this case, obviously we find that the formation of progenial colonies is facilitated by the creation of these dividing cells, which are referred to as gonidea. That is, those cells which actually get enlarged within a colony, subsequently undergo the mitotic division, and ultimately result in the formation of a new colony, which in terms of morphological features, number of cells, as well as the active components of the cells in that colony is at par with the parental generation or the parental Volvox. This completes the asexual means of reproduction. Then the next part is the sexual means of reproduction. That is where The reproduction takes place through first the formation of the gametes and subsequently the fusion of these gametes, formation of a zygote. This zygote undergoing meiotic division, forming the haploid cells, which will in turn give rise to the progenial Volvox colonies. That in turn means that in a sexually multiplying Volvox, we must have initiation which results in the formation of two types of gametes, that is the male gamete and the female gamete, and these in turn will fuse, that is referred to as fertilization, and after fertilization they will also reduce, that is referred to as meiosis, and after meiosis give rise to the progenial generation. In case of Volvox, the sexual reproduction is oogamous, that is where we have usually two types of gametes being formed. One is referred to as male gamete or sperm, and second is referred to as female gamete or the egg cell, which in turn has to fuse to give rise to zygote, and this zygote has to undergo the meiotic division. Now, usually we find that sometimes in certain Volvox we have a monoecious nature, that is where within the same colony both the male as well as the female types of gametes will be formed, and then within the same Volvox colony the gametes will be fusing, giving rise to zygote, which will result in the progenial colonies. But in some cases we may have dioecious Volvox, that is where some of the colonies will be only male colonies, some of the colonies will only be female colonies, resulting in the formation of male gametes in one colony and female gametes in another colony, where these have to be dispersed, and during dispersion they have to flow through fresh water and then fuse to result in the formation of a zygote, and then zygote has to produce the progenial colony through the meiotic division. Irrespective of what the mechanism is, whether that is monoecious or dioecious, that is where the fusion is within the colony or outside the colony, once the zygote is getting formed, it has been seen that this zygote initially enlarges in size. That is where the fusion of the sperm cell and the egg cell is enforcing the zygote, that is the diploid progeny, to actually enlarge in size, and during its enlargement it is able to actually synthesize whatever will facilitate the subsequent development, division, both mitotic as well as meiotic, to result in the formation of the progenial colony. Under normal habitat conditions, a zygote can remain dormant for about 2, sometimes about 5 hours. But if the habitat is not suitable, it has been seen that sometimes the zygote remains dormant for about 24 and sometimes even 72 hours. That is, the zygote is itself able to assess what suitable conditions will facilitate not only its division but also development of the progenial colony of the Volvox. Now, as soon as suitable conditions are seen or observed by the zygote itself, it undergoes first the meiotic division, that is where the diploid chromosome number is reduced to haploid chromosome number, and after the haploid progenies are created, each haploid generation in turn multiplies almost in the same way as we have seen in case of vegetative propagation, and gives rise to the progenial colonies, which are comparable in terms of morphological features, in terms of the number of the individual Volvox within a colony, at par with what the parental colony was. That means, irrespective of the means of propagation used, whether it is asexual or it is sexual, the parental colony is comparable to the progenial colonies, but this actually is governed by whatever genetic inheritance are owned by both male and female gametes, as well as those cells which show signs of vegetative multiplication, depicting thereby that in case of Volvox, the vegetative means of propagation as well as sexual means of propagation are actually facilitating creation of progenies which are morphologically, functionally, habitat-wise as well as habit-wise same as the ancestral generations or the ancestral parental generations are like. It means that in Volvox, whatever parameters are operative in life cycle that have been actually implanted in that particular alga by not only the habitat conditions but also whatever it has inherited from the genetic material that it has owned from its own parental generation. It means that Volvox life cycle is an important example of how primitive plant generations converted themselves from individuals initially to colonies, and from colonies to multiple colonies, and this multiple colony development does not in any way affect whatever the parental individual or parental colony has inherited from the parents in terms of its activity, in terms of its habit, in terms of its habitat, as well as in terms of its potential to compete with whatever plants, whatever microbes are living in the same habitat in which this particular plant, that is Volvox, is living. It means that the life cycle itself explains all important parameters that are either naturally operative or implemented by the plant itself, that is Volvox itself, to complete various stages of life cycle, right from the gamete formation, right from the division of parental Volvox to the formation of the colony. I hope those of you who want to analyze this Volvox would try to follow the same parameter, that is where they will first assess the colony, then assess the formation of the gonidae, formation of the gametes, formation of the zygote, division both mitotic and meiotic, and see whether the parental colony of Volvox is comparable with the progenial colony of the Volvox, depicting thereby that whatever we read or whatever we study is at par with what is naturally operative in this particular alga, that is Volvox, in the habitats in which it has a preference to go. And I hope that also in future, if you want to analyze this, you can monitor its developmental pattern in freshwater bodies, where it is very easy to not only extract it but also regularly see how variation in the density and diversity of this Volvox is. My only suggestion is that do not try to alter the habitat condition by addition of those materials who have a direct negative impact on the life cycle, both vegetative means as well as sexual means of propagation of Volvox. And in future, I hope that you will try to see what one can do to facilitate proper development, proper multiplication of this Volvox in those habitats where its color is inducing greenness to the water habitats. Thank you.